Hello, and welcome to the Chapter 14, Part 1 lecture on Mendelian Genetics. You should use the information in this lecture to complete the Chapter 14, Part 1 guided notes, which of course you should complete before you come to class. In this chapter, we're going to discuss genetics, also known as inheritance. And this is the process by which genes are passed from a parental generation to the offspring. In a previous chapter, we discussed gene expression, and we talked about how cells can turn genes on or off to control that gene expression. In this chapter, we're going to focus more on how the combinations of genes that you get from your parents can affect gene expression. Different variations of genes interact in different patterns, and so we can use this information to make predictions. For instance, we might say if a blue-eyed person and a brown-eyed person have some kids, what will the eye color of their kids be like? Incidentally, brown-eyed genes tend to dominate over blue-eyed genes. So if a brown-eyed person and a blue-eyed person get together, it's likely that all of their children will have brown eyes. But there are a number of different ways that these alleles can interact with each other, and we're going to talk about that in this chapter. In this first part of the chapter, we're going to discuss Mendelian genetics. Gregor Mendel, or Mendel, was a monk that lived in the late 1800s in what is today the Czech Republic, and he is considered the father of genetics. In those days, it was not uncommon for men of the church to do scientific research on the side, and that's what Mendel did. He was most passionate about botany, and specifically, he really liked working with pea plants. So what he did was carefully breed together pea plants, and he would carefully keep track of the characteristics of the parents and the offspring. And more importantly, he took very careful notes on all of these characteristics and how they traveled from one generation to the next. He did this over a period of years and worked with something like 28,000 pea plants during that time. And so from all of this information, he was able to divine or put together the basic patterns of genetics that still hold true today. So that's why he's considered the father of genetics. For his experiments, Mendel would choose a particular characteristic to track from one generation to the next in his pea plants. So for instance, in this experiment, he was looking at flower color. Now for this first set of experiments, he chose what he called pure breeding or true breeding plants. And a purebred plant is one that, when self-fertilized, will only produce the same characteristics in subsequent generations. So in other words, if you take one of these purple plants and you breed it with itself, which is something you can do with plants, it will only produce purple offspring. He also took plants that were pure breeding for this white flower characteristic. In other words, if you breed this white flower to itself, it only produces white offspring. And then he decided he was going to breed them to each other to see what would happen. So he took a pure breeding purple plant and a pure breeding white plant and bred them together. This first little experiment produced some surprising results. Mendel was breeding together two true breeding flowers, and this first generation he referred to as the P generation, or parental generation. He found that when he bred together two pure breeding flowers, that in the first set of offspring, which he called F1s, or first filial generation, that the white characteristic disappeared. In other words, when he bred together true breeding purple with true breeding white, he got purple flowers for that entire generation. He then decided to take two of those purple flowers and breed them together and produce the next generation, which he called the F2s. And when he did that, lo and behold, there were some white flowers in that generation. So that white characteristic disappeared for one generation, but showed up again in the next generation. He repeated this experiment many, many times, and from this he got consistent results and was able to conclude that these genes, these characteristics, are passed from one generation to the next in very specific patterns. He was also able to determine that some characteristics are what he called dominant over others. In other words, some characteristics are more likely to show up in hybrids, in mixtures of these genes, than others. Mendel worked with a number of different characteristics in pea plants over the years, and he was able to determine that many of these characteristics behaved by the dominant recessive pattern that he had seen in flower color. So he looked at all sorts of things. He looked at the arrangement of the flowers on the stems, the shape and color of the peas themselves, the shape of the pea pods, and even the height of the flower. He found that all these characteristics played by these rules. Now keep in mind, Mendel didn't know anything about DNA or genes or chromosomes or any of those modern discoveries. He referred to these different characteristics simply as characters, and he could describe how they were passed from one generation to the next. But he didn't know anything about the mechanics of why that happened. Today, of course, we do know a thing or two about DNA and chromosomes and genes, and so we can use these modern discoveries to back up Mendel's rules. They provide a physical explanation for how these characters, as Mendel would say, travel from one generation to the next. We know, for instance, that genes code for these different characters, and that you have many, many, many different genes. We also know that the genes ride on the chromosomes. They are carried by the chromosomes. Diploid cells are going to contain two copies of every gene, and that's because one of them is maternal and one of them is paternal, and they exist in these pairs called homologous chromosomes. Now, the maternal gene and the paternal gene may differ just a little bit from one another. For instance, if this little gene up here is for eye color, maybe mom give you the gene for blue eyes and dad give you the gene for brown eyes. So they're not entirely identical, and it's actually the interaction between those two sets of genes that makes you you. The presence of one gene can affect the expression of the other gene, and that can make some unique combinations. Gametes, including egg and sperm, are haploid by comparison, and that means that inside an egg or a sperm cell that your body might produce, there's only going to be one copy of chromosome 1, one copy of chromosome 2, and so on. They don't exist in pairs. Now, when your body makes that egg or sperm cell, because they're only going to pass on one of those chromosomes, that means that there's only going to be one copy of each gene present instead of two. Whether it represents the maternal gene that you originally inherited or the paternal gene, we don't know. There's really a random chance of passing on either one. And remember that because of the way meiosis works and because of crossing over, that each of these chromosomes actually represents a combination of your maternal and paternal DNA. The different versions or variations of a gene that exist are referred to as alleles. Let's take eye color as an example. The most common alleles for eye color are brown eyes and blue eyes, but there are other alleles that exist. Some people are born with purple eyes, for instance, or even yellowy looking eyes. Those are all alleles for that gene. Now, the genes are inherited in pairs, so that means that potentially you can inherit the same allele for a gene. For instance, let's say you inherited the blue eye gene from both parents. Or it's possible to inherit two different alleles for a particular gene. Let's say that this gene down here is for hair texture, and maybe one parent gave you the allele for straight hair, and the other parent gave you the allele for curly hair. You can get two different alleles for that gene, and they have to interact with each other to produce you. Individuals who inherit the same allele for a characteristic from both parents are referred to as being homozygous for that characteristic. Homo means same, and zygous refers to that fertilized egg. In other words, if we could see inside that fertilized egg, we'd see the same allele on both chromosomes for that characteristic. If an individual inherits two different alleles for a particular gene, we say they are heterozygous for that particular gene or at that particular locus. Hetero means different, and zygous refers to the fertilized egg. So it means that they are different within that fertilized egg. Different alleles have different patterns of expression. Alleles that we refer to as being dominant are always expressed when they are inherited. So in other words, if you inherit a dominant allele for a particular characteristic from just one of your parents, it's going to show up in you physically. Let's use Mendel's pea plants as an example of this. One of the characteristics that Mendel examined were the flower colors in his plants, and he was breeding together purple flowers with white flowers. He found that when he bred white and purple parents together, that the next generation contained only purple flowers, and that the white characteristic disappeared. So he declared that the purple characteristic, the purple allele, was dominant over the white characteristic. Recessive alleles are only expressed in the absence of a dominant allele. This means that they have to be inherited from both parents in order to show up physically. In Mendel's pea plants, white flowers were considered recessive to the purple flowers because white flowers showed up less commonly. Now, why is it that some alleles dominate over other alleles? Well, there can be many explanations, and it really depends on what that gene actually does, what its function is inside the cell. In the case of the pea plants, the recessive allele for white flowers tells that flower to not lay down any pigment. So the absence of pigment means that that flower is going to turn out white. On the other hand, the purple allele tells the flower to lay down purple pigment. So if that individual inherits only one dominant allele, it's still going to lay down purple pigment, and the flower is still going to turn out purple instead of white. The genes that an individual actually inherits is referred to as its genotype, and we refer to the genotype using capital and lowercase letters. In the dominant recessive system, there are three possible genotypes that an individual can have. If the individual inherits two dominant genes, two dominant alleles from each parent, we say they are homozygous dominant. In the case of the purple and white flowers, we would say they have the genotype big P, and they would end up with purple flowers. If the individual inherits one dominant and one recessive characteristic, big P, little p, we would say that they are heterozygous for that characteristic, and they would still end up with purple flowers because of that dominant allele. The third possible genotype is referred to as homozygous recessive. This occurs when the individual inherits two recessive genes from each parent. So in the case of Mendel's flowers, this individual would be little p, and would have white flowers. The genotype that an individual has for a particular characteristic occurs on the DNA, and so we can't see that. We can't see the genes that an individual actually has, although we can make some assumptions about it sometimes. On the other hand, the phenotype represents the actual physical expression of those genes. So these are things we can see. We describe the phenotype for an individual using different kinds of descriptive words or adjectives. So things like purple flowers, white flowers, tall plants, short plants, wrinkled peas versus rounded peas. Those are the kinds of terms we use for phenotypes. Now, in the basic dominant recessive system, there are two phenotypes that can occur. There's the dominant phenotype and the recessive phenotype. In the case of the pea plants, the dominant phenotype would be purple flowers. Now, that could occur because of either the homozygous or heterozygous genotype, but we can't see those genotypes. So the phenotype itself is purple flowers. The recessive phenotype in this case is white flowers. That can only occur because of the homozygous recessive genotype, but again, we can't really see those genes inside. So we would refer to the phenotype as being white flowers. I want you to take a few minutes and practice using these terms. And for this exercise, we're going to look at earlobe shape. Now, there are actually many different variations on earlobes that exist, but the two most common are referred to as the free earlobe and the attached earlobe. The free earlobe comes down and goes back up and leaves a little space between the earlobe and the head, whereas the attached earlobe goes straight down to the side of the head. The free earlobe is considered dominant over the attached earlobe. So what I want you to do is take a minute and look in the mirror and determine which kind of earlobe you have, what genotypes you might possibly have, and what phenotype you have. Now remember, if you have the dominant phenotype, the free earlobe, you can have two possible genotypes. You can be big F, or big F, little f. And that's because that dominant is going to dominate even if it shows up in combination with the recessive. And since we can't see inside your genes, we don't know which one you have. You might ask yourself, where do all these alleles, all these variations come from? Well, of course, variations on the DNA come from mutations. Mutations are those random little changes in the letters of the DNA. Now, what causes mutations? Well, we already discussed that. Mutations can be caused either by exposure to nasty chemicals or exposure to radiation, such as sunlight, or also from just the cell making mistakes as it copies the DNA. Only mutations that occur inside an egg or a sperm cell can actually be passed on. So if you do get a mutation in a skin cell or a heart cell, that might affect you, but that's not a mutation that's going to become an allele that will be passed on to the next generation. Dominant and recessive alleles have different patterns of inheritance as they travel from one generation to the next. Dominant alleles tend to show up in every single generation within a family. Let's take this family as an example. Here we have an individual that has the dominant phenotype, which means that he has at least one dominant allele, and here we're shown that he only has one copy of that allele. Even though only one copy of the allele exists in the parental generation, half of the children are going to inherit that allele, and half of the children will have the dominant phenotype. So dominant traits tend to show up in every generation. They don't skip generations. Polydactyly is the formation of extra fingers and or toes. Now there are many different causes of polydactyly, but the most common forms are caused by a dominant allele. So this is a condition that can be inherited and can be passed down from one generation to the next, and it tends to not skip generations. So if it exists in a family, it tends to show up at least once in every generation. Here you can see some examples of polydactyly. Here we have a person that has an extra finger here. This is also shown in cats. Cats can have polydactyly, and you can see extra little toes on this kitty cat. And look at all these extra little toes that this person has. Now, polydactyly is also an interesting example of a dominant allele because it's not actually very common. I mean, how many people do you know that have extra fingers or extra toes? So I put this here to remind you that dominant alleles are not always common. They tend to show up when they are inherited, but they're not always the most common allele that exists. Polydactyly is also an important example of a dominant allele because it doesn't really affect the function of the individual that much. I mean, having an extra finger or some extra toes might affect how the hands and feet work a little bit, but it's not going to limit the lifespan of the person, and it's not going to necessarily affect whether they can have children or not. So a person with polydactyly is usually perfectly capable of passing that gene on to the next generation.
City couple is beaming tonight. They're the proud parents of a healthy but incredibly rare baby boy. See, he has 6 fully formed and functional fingers and toes on his hands and feet. His parents asked KTVU Health and Science editor John Fowler to tell their story. John?
You really need to see these pictures. It's called polydactyly, extra digits, not an uncommon genetic trait, but Bay Area doctors tell me so far they've never seen a case so dramatic. Born at San Francisco's St. Luke's Hospital 3 weeks ago, baby Kamani Hubbard seemed so perfect at birth, no one noticed.
Nurses and doctors, it looked so normal. They couldn't tell. They told me he was 6 pounds in good health, and that was all they said.
But his dad noticed this spectacularly rare case of polydactyly. 6 perfect fingers on each hand, 6 perfect toes on each foot. The general trait runs in his family.
Some family members have had the 6 fingers, but not completely developed, but not the toes.
In fact, Hubbard himself had nubs of 6th fingers removed as a child, as these non-functional digits routinely are. But this case, doctors say, is so vanishingly rare, so functional, it's not a deformity to be discarded.
It's merely an interesting and beautiful variation rather than a worrisome thing.
St. Luke's pediatrician Michael Treese.
I would be very tempted to leave those fingers in place. I realize that children will tease each other over the slightest things, and having extra digits on each hand is more than slight. But imagine what sort of a pianist a 12-fingered person would be. Imagine what sort of a flamenco guitarist. If nothing else, think of their typing skills.
Kamani's mother says she hasn't had time to absorb this yet. His specialness didn't even show up on prenatal ultrasounds.
I heard nothing until I gave birth, so I'm still in shock, kind of.
I just want him to just, see what greatness will be in store for him.
One current big league pitcher, a modern blues guitarist, and several remarkable figures in history have all had polydactyly. Now, this little guy may, at the very least, help others grasp the importance of embracing differences. Reporting live, Health and Science Editor John Fowler, KTVU Channel 2 News.
Progeria is another condition that is caused by the inheritance of a dominant allele. So individuals that are either homozygous dominant for this characteristic or heterozygous will have progeria. Progeria actually affects the cytoskeleton inside a cell. And so remember that the cytoskeleton helps give the cell shape, it helps move stuff around inside the cell, and it gives the nucleus its shape as well. Here we have a normal nucleus supported by normal cytoskeleton. And here we have the nucleus of someone that has progeria. And you can see that because the cytoskeleton is deformed, the nucleus is deformed, and that can drastically affect how a person performs transcription and translation. So people with progeria have a lot of difficulty in making and regulating proteins. And remember that proteins run the cell. They do all the important jobs inside the cell. So people with progeria have difficulty with cell division, with healing, things like that. And this can result in premature aging. Now, by premature aging, I don't mean you get a few wrinkles a little bit early. No, this shows up really early. Most individuals with progeria have symptoms by the age of 2. Things like hair loss, organ dysfunction, wrinkling of the skin, and so on. Now, unfortunately, most of these individuals don't live to reproductive age, so the oldest known cases of progeria died in their early 20s. So it's unlikely that someone with progeria would be able to pass this gene on to the next generation. So this is an interesting example of a dominant allele because although it can be passed from one generation to the next, it doesn't tend to be retained in families. It doesn't tend to travel through multiple generations. And that's because it's caused by a germline mutation. To remind you, a germline mutation is a random mutation that pops up in an egg or a sperm cell. While dominant alleles tend to show up in every generation of a family, recessive alleles are quite a bit sneakier. Recessive alleles can skip generations. Now, why is that? Well, it's because the recessive allele is only physically expressed in homozygotes. So that means that an individual has to inherit it from both parents in order for it to physically show up, in order for them to have that phenotype. Now, what if you're heterozygous for a trait? You have the dominant from one parent and the recessive from the other. Well, that would make you a carrier. That means that you carry the recessive gene, but it doesn't show up in you physically. However, because you carry that gene, you can pass it on to your children. Let's look at an example. Here we have a couple, and both of these individuals are carriers. They are heterozygotes for that trait. With two heterozygotes having children, the children have a 25% chance of being homozygous dominant, a 50% chance of being carriers themselves, and only a 25% chance of physically expressing that recessive. So because carriers are present, it means that neither of these parents showed the characteristic, but it can show up in their children. So it can skip generations. An interesting condition caused by a recessive characteristic is albinism. Individuals with albinism have difficulty making this pigment melanin. Melanin is the dark pigment that's produced by your skin. It builds up as you tan and makes your skin darker, and in general, the more melanin you have, the darker your skin tone tends to be. People with albinism don't produce melanin, and so the skin, hair, and even eyes can be very pale in color. Interestingly, individuals with albinism will have pink eyes, and that's because the front of the eye, the iris that normally has color in it and normally has melanin in it, has no pigment at all, and so the light shines through and bounces off the pink retina in the back. So you're actually seeing the retina on the back of the eye. Now, individuals with albinism have a lot of difficulty with sunlight because melanin protects you from sunlight. And they can have visual problems as well because they can't control the amount of light that's going into their eyes. Animals with albinism are most commonly seen in zoos, and you can probably guess the reason for that. Look at this rattlesnake. If this rattlesnake were crawling around outside in the leaves, it would be pretty easy pickings for a big bird. But you see these animals in zoos a lot because they're pretty easy to breed for, and they look interesting. They draw a lot of attention. All right, you guys, you're finished with this part of the chapter. Make sure you thoroughly complete your guided notes, and it's a pretty good idea to practice with some of these terms before you come to class too. Things like heterozygous, homozygous, dominant, recessive, phenotype, and genotype, because we're going to work with them a lot.